The overall goal of this procedure is to determine tolerable fatty acid and cholera toxin concentrations that do not negatively influence cell survival. This is accomplished by first growing the appropriate type of cells. The second step is to incubate the cells with varying concentrations of the 3 fatty acids to be tested, or with the cholera toxin. After 24 hours, cell survival is assessed using an MTT assay. Ultimately, spectrophotometry data from the MTT assay show optimal concentrations of fatty acids and cholera toxin with which cells can be treated without significant reduction in survival.
The implications of this technique extend towards the prevention of cholera infection because vaccinations are not always successful.
Moose musculus macrophages from BALB-C mice will be used for all cholera toxin determinations and for larger scale concentration determination of fatty acids. The cells are propagated in either of 2 media: DMEM with L-glutamine, completed with 10% fetal bovine serum and 1% antibiotic antimycotic, or RPMI 1640 base media, completed with 10% fetal bovine serum, 5% L-glutamine, and 1% antibiotic antimycotic. For this protocol, grow the BALB-C cells in 75 cm square Corning flasks with vented caps at 37 degrees Celsius, 95% air, and 5% carbon dioxide. For fine-scale fatty acid concentration determination, use human intestinal epithelial cells propagated in 75 cm square Corning flasks with vented caps in Hybricare media completed with 10% FBS and 30 nanograms per milliliter human EGF.